Okay, so we're getting ready. Sorry for the delay, but we were having some technical issues. So this is the final part of today's workshop sessions. And as I was mentioning, if you attended the previous workshop, both the previous workshop, the one in the morning, and this roundtable, they have been supported by a NSF grant. And we would like, first of all, to thank the NSF for supporting the activities for the whole day. And if you have been following both the morning and the afternoon workshop, this final hour is going to be a slightly different format. We envision this to be a roundtable, an open conversation with the panelists that they're going to introduce themselves shortly, and then you, the audience. What we did in here, we put together a group of individuals that they have expertise in different fields when it comes to tissue that they use for their experiments and for their application. So we go from plant to mammalian to invertebrate and so on. And the idea is to really see if there is a common thread among the different disciplines when it comes to media formulation and associated complexity every time that we need to either develop a new media or optimize an existing media. So now, without any further ado, I'm going to have the slides going one by one, and then I'm going to ask whoever is on the slide to introduce themselves using the microphones, and then we're going to open the conversation with the floor. So the first person is Cindy.
Hello, everyone. My name is Cindy Goodman. I retired from USDA a couple years ago. I'm now working part-time for a small company that focuses on cryopreservation. But really, my background is insect cell culture, and I've worked for the past 30 years helping develop cell lines and then characterizing them. So I've been quite passionate about working with insect cell lines and have really enjoyed the process. But one thing that we've discovered, I mean, of course, the purposes for why you're developing cell lines does change over the decades. And the past couple decades, a real focus has been on developing in vitro models that mimic in vivo systems. And for us, working with insects in the insect cell culture world, that has been our major challenge. We can develop cell lines from many different tissues, including nervous tissues, midgut tissues, aorta tissues, all those different tissues from many different species. But the question is, are they behaving similar to those same tissues in vivo? And so in a lot of cases, we're not finding that to be true. So we're really in a new position now. We've really become experts at establishing cell lines, but we need to move on from there. And really, I think the key is in optimizing the media for each of these cell lines so that they will continue to behave as if they're within the in vivo system. So that's really has been a more recent goal, and that's really why I'm part of this panel discussion, because we really know the value of highly optimized media. Thank you.
Perfect. Thank you, Cindy. Now, next is Naomi.
Hey, y'all. I'm Naomi Sonic. Y'all hear me? Okay, there we go. I work at WashU here in St. Louis, and I run the Digestive Diseases Organoid course. So I work maybe 10% with mouse GI organoids and about 90% with human GI organoids. I receive patient samples from patients that have Crohn's, ulcerative colitis, colon cancer, liver cancer. Then I establish these 3D epithelial organoid cell lines from those. And we have a huge biobank of said cell lines. Researchers at WashU can then hire me to set up experiments for them with said cell lines. And then I essentially either 3D or 2D set up these plates and hand them over to the researchers to do whatever it is that they need to do. One of the big goals with the human samples is to do patient-based studies or like treatments on these cell lines. So let's, you have a patient with Crohn's disease, let's treat your cell line, see how your organoid line reacts to treatment, and go from there versus putting you through all the different things. In regards to media, the organoid core has, I make the 50% LWRN conditioned media, which expresses Wnt, R-spondin, and noggin, which are necessary for maintaining that proliferative state of these human organoid cell lines. I also have separately R-spondin, separate noggin, and separate Wnt medias, and all of these kind of differ depending on your organoid, like on your organ that you're working with. So liver has different requirements. Your GI has different requirements. And then also like various other reagents and inhibitors, or like the matrices that you grow these organoid lines in. And of course, I also do like the QC testing for all of these medias.
Perfect. Thank you, Naomi. Next is Nathan.
Hi everyone, I'm Nathan Vo. I'm a faculty member at In Vitro University from Ontario, Canada. So I've spent the last 15 years doing fish and amphibian cell cultures. I have developed and characterized many cell lines from fish and amphibian in the past, probably over 40 by now. I have worked with different life stages of different fish and amphibians. Current research in my group focusing on fish hemopoietic cell cultures. So I'm trying to grow fish dendritic cells, which are known to be hard to grow. And I have some success recently. So I'm open to any question that you may have. If you want to develop fish cell lines or frog cell lines, I'm happy to help.
Okay, thank you, Nathan. Now we have John.
Good afternoon, John Harbell. My background is in vitro toxicology, particularly predictive assays for topical toxicity in humans. Those started out as keratinocyte, fibroblast-based cultures, and eventually we found that we really needed a three-dimensional tissue construct models for those work. My first laboratory was in the Army, and the Army has a purchasing system that is about as responsive as the worst university you've ever heard of. You know, they're good at buying tanks, but if you want culture medium in a timely fashion, it doesn't work. So I would buy 10x medium, and I would make my own tissue culture grade water. Well, I found out very quickly that those deionization columns do not make tissue culture grade water. So, or if you have a little filter on the end of your distiller, that doesn't make tissue culture water because the bacteria grow in there and the endotoxin produced is quite high. And some cells, like L929 cells, hate endotoxin. So the message of that is if you're going to be developing medium, start with the best water you can buy. You're going to distill your own, autoclave it before you store it. Thank you.
Thank you, John. Shirley?
Thanks, Piero. My name is Shirley Pomponi. I am very recently retired from Florida Atlantic University, but seem to be busier than ever. My area of interest, expertise, passion is marine sponge cells. Took us about 30 years, but we finally developed a sponge cell line in 2019. Our group was successful at doing that, and we've got another cell line that has, I think it's continuously dividing. I'm not sure it's immortalized or not. The problem with sponge cells, and probably any marine invertebrate, is that they're not like mammalian cells. The methods that have been developed for mammalian cells, they've always got to be modified, whether it's, you know, your medium, of course, because the cells are growing 10 times the salinity as mammalian cells, for example. Not only that, but there are 10,000 species of sponges, so we're not dealing with just, you know, mouse, human, whatever, insect, whatever. We're dealing with lots of different species, and we're very lucky if we can get a species that doesn't exhibit individual variation. So we may not only have variation between or among species, but also within species as well. And following on with what Cindy said, our original goal when I first started doing this so many years ago was to develop an in vitro synthesis system for production of bioactive compounds that are produced by sponges. They're a very prolific source of bioactive materials for pharmaceutical applications. But as the program has evolved over the years, we've really become a lot more interested also in biodiversity conservation, being able to habitat restoration, do 3D modeling, and so on. And I would say that probably our biggest challenge right now, I mean, I can't believe I'm actually even saying this, we have actively dividing cells, and they divide fast. So we're really happy about that because it took a long time to get there. But now our biggest challenge is making sure we can get those cells to differentiate. So I'm really interested in what you all have done in terms of expansion versus differentiation, because right now that's kind of a bottleneck for us. There are some sponge cells that will aggregate and differentiate really rapidly. Unfortunately, the very first person who did this back in 1907 lucked out and got a sponge that aggregated and differentiated and formed a nice sponge that had canals and everything, but that's really an anomaly. That's an outlier. That doesn't usually happen. So I'm really interested in hearing some of your advice or guidance on developing different media that will support differentiation.
Perfect. Thank you, Shirley. Now we have Greg and Todd. Not because we were running out of space on the slide, but it was just, I don't know why we ended up to put the two of you together.
So my name is Boy George.
Actually, I was challenged because I chose the title and I was challenged how many people had a reference to the Culture Club. You do.
So I'm Todd Jones, and I have not retired yet, although I probably should. I've been 35 years in industry the entire time, and for most of that time I've worked on your favorite row crops: corn, soybean, a little bit of sorghum and wheat, cotton and canola. Dabble a little bit in things like millets and cowpea, and for a brief period of time I also worked on trees, which is a completely different system and takes too damn long if you ask me. The one thing that, and kind of to Shirley's point, many of these systems rely on somatic embryogenesis, this very cool thing where plant cells are totipotent, or can be, and a somatic cell can turn into an embryo. And we would love to be able to recapitulate zygotic embryogenesis. But oftentimes we don't. We get something that's just good enough that it becomes a somatic embryo. It has a shoot pole and a root pole, and it'll turn into a plant, and that's fine. But I think it'd be wonderful if we could develop media that would actually recapitulate zygotic embryogenesis completely.
Great. Okay. I'm Greg Phillips. I've been retired 3 years from Arkansas State University. I noticed the program. I thought I'd put my title in as emeritus professor, but I deliberately left out Arkansas State University because they don't believe that emeritus professors deserve to have a university email address. So I'm pissed at it. Anyway, I'm unaffiliated now. I've been an academic my whole career, 40 years at New Mexico State University and Arkansas State University. I've worked with soybean and rice, which you might have heard of, but I've also worked with chili peppers, onions, tomatoes, cactus, pine trees, hardwood trees like pecans, willows, all kinds of plants. I've done a lot of basal medium manipulation for those different species. That's probably where my greatest expertise lies. I'm ready to pass it on, Piero.
Okay. So, Bill, I think you're the last one.
Hi, I'm Bill Gordon Cam, and I've been working in predominantly maize transformation since 1994. And when I started work in this area, people had already been working on plant tissue culture media for decades. And, so all the modules for putting the media together had pretty much been established. And we were just taking, known mixtures and adding and subtracting. And then we started adding and adding and adding over the years. So our media has continued to get more complex, and it works, but it's really frustrating. And then I got tired of all that complexity, and I thought, okay, I want to use genetics to stimulate growth. So I kind of switched gears, and I started looking for genes that would stimulate cell division or stimulate morphogenesis, and try to use those to do our selection for transgenics. And we've been really successful, but we're still using that same really overly complicated media. And so one of the areas that I'm interested in now is we have genes like Wuschel and Baby Boom and others that will stimulate somatic embryo formation or different combinations that will stimulate shoot formation. If we use these new genes now as, you know, kind of the crux of our system, you know, what can we remove from our media to simplify the media and maybe even start from scratch and rebuild it? So listening to the presentations this morning on how to build a media from scratch was kind of exciting, but also kind of scary.
Okay, so thank you everybody for the introduction. I just want to remind all of you that a panel like this, with such a diverse level of discipline, is possible because of what is SIVB. SIVB is a society that really goes from plant, invertebrate, fish. So you heard the different field of application in here. Now, it's also interesting to hear recurring topics, like you, John, mentioned water, and just in the afternoon workshop, Ken Torres was mentioning how important water it is and how a lot of people don't even realize the importance of water. Now, I'm going to open the floor for questions, and you heard how pretty much everybody reminded us that it's always important to begin with the end in mind. What do you really need your media to do for you? In the case of Shirley, was important, for example, for her to start to see the vision. Now our next end is how can I move forward from that simple division? I'm opening the floor for the questions. There is a microphone in here, and I can grab this microphone if someone is on the other side. So again, this is envisioned as an open conversation, roundtable. If you have questions, challenges that you are experiencing in your current experiments, or if you have specific questions that you want to ask to any of the panelists about how they overcome a challenge, how they proceed when they need to develop a new media, that's kind of the idea how we want to proceed in here. And there is always that awkward moment of silence, and then you have the first question, and then you go over time.
But let me just say that first, whenever my experiments go bad, I always blame the water.
Mine's more of a comment than a question. If you can hear me, I won't get up and use the mic. I'm pretty loud. When I first started in vitro, we had a cell line that had been established in Boston. And the PI brought it down with him to Long Island. And he had always used powder media, which we reconstituted with good water in the lab. And the cells were miserable until he started importing water from Boston as well.
That confirms the importance of water. Yes, we have a question over there. I can come over. We want the question to be recorded on the video. This is going to be recorded, by the way, and then it's going to be posted on the YouTube channel of the Society.
I'm Kelly Chapman with Viscon, and I would like to ask, if it hasn't been covered already, can you talk about what water quality analysis you would do besides, say, pH or some of the obvious ones? What should be done?
Well, first, you should never use water from Stony Brook. The obvious things are minerals and so forth, but even clean water that comes out of a ion exchange column still can pick up endotoxin, for example, from bacteria that grow downstream in that resin bed, particularly at the end, because you've taken all the chlorine out, so there's no disinfectant. And so that's one of the things. You also want to make sure there aren't any, I'll call them toxins for want of a better word, in the local water supply. Some water systems use not chlorine, but different forms of chlorine, which are much more stable and toxic. And so, for example, if you raise fish, you want to be aware of that because it'll kill your fish. It'll certainly kill your cells. So what I found to be most successful is to buy tissue culture grade water from suppliers, because then you have it and you know it's been tested and clean. You also want to make sure that it's sterile, and so when you open it, you want to keep it refrigerated and keep it sterile. I mean, treat it as if you're dealing with culture medium as opposed to just water, because even low-level bacterial contamination will proliferate even at 4 degrees. So just treating it like something special is worth the investment. And don't do it in the Army.
I actually have two questions, but I'll start with one and you can kick me out and I might get to the other. Actually, more for the animal people. How much demand do you see in using alternative sources for some of the media reagents that are non-animal? And do you do that? Is there a market for it? What do you see as a driver that might be something to be filling? I know there's companies that are doing that.
Well, Alan, just from the insect point of view, I mean, I'm sure you know, and many other people know, there's a number of different insect cell lines that are used to produce biomedicals. So, in especially those cases, we want animal product-free media. Now, for the kinds of experiments that, you know, ag chem companies may need, such as this whole idea of using cells in pesticide discovery research, that may not be as important. But certainly if it's going to be used in some kind of biomedical application, that would be an important reason to have that available. Does that answer your question?
And the other thing we have to deal with is that certainly in the marine invertebrate cell lines, you have a lot of contamination. And right now we use antibiotics. So obviously if we start trying to produce some compounds for pharmaceutical applications, we have to be able to culture those cells without antibiotics.
There was a talk this morning on producing fish cells for food. And one of the points, and I don't remember the name of the material, but it's derived from the purification of rapeseed oil, the protein. And apparently it makes a really good substitute for major components of fetal calf serum. So that's the kind of thing that a particular industry might develop for their purposes. But again, going back to, pardon me, biopharmaceutical, or even my own field in vitro toxicology, there's a move to get away from animal products. And fetal calf serum is also so very, very expensive. So anything you do to get the cost down would be helpful. So just an example.
Second one? Okay. The second one's more about application, so if it's not appropriate, don't answer. But how, and again, more in the animal field, how are you looking at physiologically relevant levels of response to when you're using cells to give you a readout on something? And how might that actually, how may media elements interact with that to confound that analysis?
I think it's the question that has been going on for many years. So obviously, nowadays we do have other more complex cell culture system, like intersystem, where you can do like layers of different tissue to try to mimic, at least to the best of our abilities, the in vivo response. But there are differences, and I think it's important for us as in vitro biologists to ask why there are differences, and what can we do more to try our best to mimic the in vivo systems using cell cultures.
And you know, what are you really looking for in case of toxicology? You're looking for an upstream predictor of a downstream event that you measure, say, in the animal. And let's say eye irritation, because that's been a favorite topic an interest of mine. What is it that makes that eye turn opaque? What is actually happening in the eye? And it wasn't until 20 years ago when Procter & Gamble funded research to actually ask that question. And from that point, then we had a way to understand the physiology of eye damage so that we could actually start to model it in vitro. And it turns out the modeling we needed was not single cells, but complex tissues, regenerated human tissue, or human cell-based tissues, cornea, for example. And secondly, the importance of consistency in the model, what the regulatory people call the test system, whether it's cells or tissues, but they really have to be consistent so that the endpoint that you measure, whether that's cell viability or cytokine expression or whatever it might be, is based on the same relative number of cells being exposed to known material, quantity of material, and then probed the same way each time. That is, whether you use Alamar blue, which you actually can't do in the three-dimensional cultures because it only measures the bottom, but MTT, for example, using it the same way, measuring it the same way, checking to make sure that your test material doesn't activate Alamar blue or MTT directly, which some of them do, and then you can start to have a prediction. And finally, you have to have a prediction model. What concentration or what time to toxicity or how much toxicity is going to be predictive of some exposure under standard conditions in vivo?
Great, thank you.
Is that fair?
I'm going to add a little bit to that, if you don't mind. So for the media that I generate in-house, it's also very available to all the researchers at WashU, so I need to make sure that it is very reliable and very consistent. Postdoc and we put out a publication on how to QC test this media several years ago. So one of the assays, for example, that I use is called cell titer glow assay. It's a luminescent assay, and it's going to read the ATP activity in those cells. And so essentially I use like human GI, mouse GI lines, and then I feed them with my previous batches of media, my current batch of media, and I compare, and then I also have a condition where I've differentiated those cells. And so I'm, like he mentioned, the consistency, so I'm checking batch to batch to batch. So if 4 times a year I generate these 16 liters of media, I need to make sure that every single time they're performing as they should. And then I also use PCR analysis to check for stem cell markers, Wnt activity, and then also compare that back to differentiation, which I don't want because we want our cells to be maintained at this stem cell state until we tell them to become adult colon or adult small intestine. But we want them to be maintained as a stem cell. So I QC test my media to make sure that it actually consistently has that sort of environment.
Hi, so I'm Greg Gorologia from Oregon State University. One of the things that I feel a little bad about is the amount of waste that I generate in the lab. I can probably fill up like a Honda Civic with the amount of Petri dishes and media that has to get autoclaved every week. And, occasionally I'll use glass Petri dishes for pilot experiments or something, but I was wondering for all the panelists if you found any laboratory practices that reduce your waste, or be it plasticware or otherwise.
Yes. Yes. So I'm German. We love to recycle, if you've ever been to Germany. So I hate wasting things, and I've tried to carry this over into my organoid core. And WashU has this program that is also, it's called Green Labs. So they're trying to encourage labs to have more green practices. So maybe purchasing your -80s, whatever it is, being more energy efficient, unplugging equipment when you don't actually need it. And then also there are a lot of, there's companies that actually produce plasticware, so your pipette tips, your serologicals, all that kind of stuff. That's a little bit, obviously, in my case, everything needs to be sterile, filtered. I can't use unfiltered tips, so that kind of limits my options on that. But Eppendorf has a line called bio-based. So all of those pipette tips, serologicals, et cetera, they are more eco-friendly, think carbon neutral, all that kind of jazz. And I think they're made from oils, like plant oils, so they break down more quickly. So things like that. There are definitely reagents and things out there that you can purchase that are a little bit more friendly.
On the other side, when I was a graduate student, the laboratory I was in ran out of money. And so we were rewashing disposable glass pipettes, sometimes losing all the numbers off them, and we just use them for transfer. But the point of this message is that we got a mycoplasma contamination, and fortunately it wasn't my work, but other in the lab were using the pipette to feed one vial, go back in the media bottle, get another load, put it in the next vial. That is a recipe for disaster. And those pumps are also a recipe for disaster, because you have the risk of transfer for aerosol or otherwise of not only contaminants like mycoplasma, but also cells. And so your HeLa cell contamination in the laboratory is part of that. So just conservation is great, but don't put your whole program at risk. Is that a fair decision?
Nathan, yes.
I have had the opportunities to work in different labs, fish, amphibians, human cell culture lab, and what I found was that when I worked in a human cell culture lab, I was told that we only use tissue culture vessels once and we throw out. We do not recycle, we do not reuse. Because especially at the time I was in that lab, we were working on what we call a bystander response assay for radiation biology. And the cells are very picky. You have to patch them new every day before you can use them. But then when I worked with fish and amphibian cell cultures, we reuse tissue culture flasks many times. Obviously, we keep everything sterile. So I think it just depends on the research needs. I also know that there is a startup company out there trying to produce biodegradable tissue culture flasks. I don't remember the name, but I know there's a company out there that's trying to do that. So that would be something interesting to see, and I'm sure that we're going to have to test with different separator systems to see how reliable they are and do they produce same responses compared to the traditional approach.
Well, and to make sure your cells like the substrate. You know, keratinocytes in 40 years ago loved Corning and CoStar plates, but they hated Falcon plates. What did the Army buy? Falcon plates. So you had to coat them with something just because these are proprietary treatments of the surface of the tissue culture vessel, and so they're not uniform across companies. So whatever the cell likes is what the cell should get, I guess is the point of that.
I've been using the same plates for over a decade.
Ah,
Same brand of plates.
yeah.
Just to follow up on the question, if I may, at the turn of the century, we were working for a biotech company that was rumored to have used the most plastic petri dishes in British Columbia. There was a construction asphalt company that would take our lids, grind them up, and incorporate them into the roads. They couldn't use the bottoms because they had medium, but they could use the tops. That was a nice way to dispose of it and get them recycled. Unfortunately, I don't know whether they lost their subsidy, but they went out of business. My question may take a bit of a right turn, particularly for Cindy, Naomi, and Nguyen. I work with plant genetic resource conservation. I work with clonal crops. We maintain long-term material in vitro. My biggest obstacle and worry is somoclonal variation and epigenetic changes. If you're developing cell lines that are constantly growing, constantly replicating, and you're using them as models, how do you avoid getting somoclonal variants and epigenetic changes that will alter your results?
Well, that is a really good point, Dave. And something we have noticed over the years is it depends on what the endpoint is you're looking at, because the endpoint can actually be dependent on passage number. You can get a different result using a different passage number, but it seems like different systems, in some cases, it doesn't matter. So like originally, a lot of cell culture worked with insect viruses. We were looking at studying and producing them in vitro. And so in some of those cases, it didn't matter what passage your cell line was, they would keep producing the virus. But I've heard from people like Bryony Bonning that in her hands, that hasn't always been true with some virus cell culture systems. So it really depends. But one thing I should say, that especially if you're looking for something more in vivo-like, if you want to call it that, usually it's best to use an early passage number. And that is when it's just adapted and is growing fairly well. It will produce more properties that are in vivo-like, that, excuse me, as the cell line adapts to being in culture, all of a sudden the phenotype really will change, and you might lose certain receptor types that you were looking for. And so in those cases, you really want to use as low of a passage number as possible, so you're maintaining a lot of the in vivo-like properties. That's one of my observations.
I agree with that. So for like with the human GI lines, the best practice is so my goal is to freeze them down at the lowest possible passage so that from there they can be expanded and used for whatever experiments, et cetera, still within a low passage range. And so it's always encouraged to not really take them farther than X number of passages and work within that range.
And I think that not just the passage number, also the medium types. So I believe that because of fish amphibian systems, we use different medium types. For example, L15, DMEM, and at least from what I have seen is that in terms of proliferation, morphology, gene expression, they are different with different just basal media types. So what I've been trying to do is to work towards more, because nowadays we have RNA-seq, which is more still expensive for my kind of work, but I think it's more affordable nowadays compared to in the past, because I want to do SC, ATEC, RNA-seq, and even single nucleus RNA-seq. So I think that could be something that we could try moving forward, but it does require dollars, research dollars. And that's, especially for us who do not work on mammalian or human systems, research fundings are limited. So we do the best we can, but definitely something that if everyone in the audience can work towards that goal, that would be great.
And Dave, I know you know this already, but I think keeping the media as simple as possible. Right, exactly. But I have the bumper sticker. SNPs happen, you know. So, what do you do? You know,
Yeah. How many people use white light in their hoods? The fluorescent tube, what color is it? White?
Cool.
Okay. Cool. I don't care if it's cool. There is a cell line called L9521. It's a lymphoma cell line. It's used in gene tox assays. And it uses Fisher's medium, which is very sensitive to light. And so if you use that, you can measure, using the gene locus that's used for measuring genetic toxicity, to measure the spontaneous rate of mutations in that cell line, which are hellacious. And if you use white light, you will see, one, that the mutation rate goes way up, two, that the media goes downhill pretty quickly. So it's just a thought. A yellow sleeve over your fluorescent tube will reduce the blue wavelengths, which is what the critical element is in this thing. It's actually not just folktale, it's demonstrated. So.
In plant transformation, transgenic plants or gene-edited plants are generated from two-part way, somatic embryogenesis or direct shoot organogenesis. which pathway is more important and why do you think that pathway is important? And do you have any strategy to specifically induce somatic embryogenesis or to specifically induce de novo shoot organogenesis?
Take it, Bill.
So the answer is crop specific. But I think in my mind, I would break this down. You look at crops like soybean, cowpea, they transform very readily around the apical meristem. And for transformation at the apical meristem, shoot proliferation, releasing other axillary buds and actually proliferating new shoots, along with selective agents like spectinomycin, can be extremely beneficial. All direct shoots. For cereal crops like maize and sorghum, direct shoot formation isn't as useful yet, so somatic embryogenesis is the preferred method. And the difference between those two, I think, can be interesting when you start thinking about problems like somoclonal variation, because so length of culture, length of cell divisions going through culture, but it's not just culture. If you could do the culture without hormones, I don't think we'd have such a big problem. But I think when you start adding things like 2,4-D, it adds a lot of stress to the system. So I think duration of exposure to 2,4-D is a major culprit of somoclonal variation. And I think there are a lot of crops that tolerate more of a cytokinin shoot proliferation method. So that's going to probably be the much preferred method.
Yeah, let me clarify again. I think I'm not talking about direct shoot organogenesis. I'm sorry. Yeah, direct shoot organogenesis from apical meristem is usually chimeric and we do not want. But yeah, when we induce K-line from H-plant, yeah, it can go through somatic embryogenesis or de novo shoot organogenesis, and both pathways are okay. So I'm talking about these two pathways, not direct shoot organogenesis. I'm sorry for confusing.
Okay, so you're referring to, for example, like the pathway where Arabidopsis roots will form shoots. Is this what you're?
What I'm talking about is, yeah, we can induce somatic embryogenesis, and also we can also get the plant through direct, not direct, de novo shoot organogenesis. So both pathways, they start from K-lines. From the K-line, they transform into somatic embryo. Then they go through somatic embryogenesis and they generate new plant. For de novo shoot organogenesis, from the K-line, they form shoot and they deploy into shoot, and then they are transformed to, they are moved to root induction media, and we got the whole plant. So.
Yep. So I think the argument going through one or the other, if you're starting with callus, I think the argument still applies. I think de novo shoot formation, leaning more towards using cytokinins instead of auxins. I think there are some crops that just go through that pathway much easier. And if you can do that, if you have a crop that is amenable to that, I think it's going to be less stressful.
If I can chip in, I can't remember the specific papers, but in the ancient literature, 1970s, 1980s, that kind of ancient literature for plant tissue culture, there were companies that were heavily involved in micropropagation of various plants, and appropriately, they would not reveal the details of their regeneration systems, but they would reveal that in general, they found direct shoot regeneration to have fewer mutations than the somatic embryos. But if you start looking at papers on a specific plant species that is propagated either by somatic embryogenesis or de novo shoot regeneration and then rooted, if you look in those cases, I'm not sure that there's a general conclusion. I think it depends on the species and the specific culture regime used. Not all regimes use 2,4-D, for example. And so I agree 2,4-D is one of the big culprits, but not every auxin is mutagenic. IAA is not known to be mutagenic unless you use it in absurd amounts, like, the level of sugar that will cause you to die, that kind of absurd amount. So, I don't know which plant species you're talking about, but I think you would probably have to do a study to determine, unless studies have already been done in that species, as to which would give you the more reliable true to type propagules. And if you're after mutations, the other one will give you more mutations, most likely.
Right. I agree, Greg. I think that's a good summation. I think it depends on the species. And it sounds like you can get both either somatic embryos or de novo shoot formation using the same hormone regime, I'm assuming. And so it may not matter necessarily. It may come down to which is more efficient, right? I think
John Feiner would say that probably they were all somatic embryos, but some of them just grew the shoot out first.
Exactly. And the other thing is, if somatic embryos tend to produce both the shoot and the root at the same time, so you may save a step. You won't have to do the root formation later, so it could just be a simpler process for you.
Hi, my name is Jacob Zobris, and I have a question about just the complexity of tissue culture and how something that may have occurred at the very beginning of our tissue culture protocol can have a great effect on the ending part of our tissue culture protocol. And how do you kind of unravel what's going on when, say, something in your germination phase is affecting your regeneration phase when all your callus actually looks the same? Does that make sense? Or
If you were asking this about human organoid culture, I'd ask you how long you have to actually listen.
Oh, okay. Yeah, I deal mostly
talk about that forever.
with plants, but I've noticed where sometimes my germination media that I used to actually germinate some of my plants on, that step actually greatly impacted whether or not I was able to regenerate plants later on. And so even though there's quite a few steps in the middle of that, how do you go about actually understanding kind of what's going on in the middle there? And
It's the water.
It's the water? Okay.
That could also be just a basal medium effect. I mean, most plants do not need a full-strength MS medium to germinate. In fact, most plants' seeds will germinate better in either 1/10 or 1/4 strength Murashige Skoog. And you don't even have to use Murashige Skoog. I mean, you can use greenhouse watering recipes. You don't have to use a complete nutrient medium for seed germination. Most seeds have most of the nutrients they need to germinate. So, but if you overload it, I have seen a case where seeds on a basal medium with no hormones developed callus instead of germinating. So that implied to me there's way too much in that medium. It's stimulating a cancerous or tumorous kind of response.
So when
because the one with auxin, you already gave it a signal to do something. And the other one had no signal to do anything in particular. So they just formed callus, and you're right, generally the calluses will look the same, but the set with the auxin from the very beginning had a very different hormone signal than the one without it. So it's no wonder to me, yeah, hormone effects can be applied months in advance of seeing the ultimate result, depending on the plant system you're working on.
And I guess my question is, how do you really unravel all of that and really?
By making mistakes and then figuring it out.
Learn by doing.
My favorite mantra is, I had a student in my lab. Oh, I made a mistake. Oh, I made a mistake. Boy, I learned a lot today.
We've all been there.
I should be a genius.
We have one last question because I think we're getting close to the end. Go ahead.
Yeah, I have a different question about the complexity of media and preparation formulation. So about the morphogenic genes that you told that the baby boom and Wuschel, these two morphogenic genes together work fine most of the serial system. And both the genes, they have same efficiency and transmission for the dicot. If not, what is the region? And the second question is that, is there any other morphogenic genes really they are working for dicot as well?
So in our experience, so this is a little bit off of media, but in our experience, Wuschel and Baby Boom work for a wide range of species within the Poaceae, but that particular combination in our hands has not worked well for the dicots that we've tested. We have other combinations that have worked well in dicots, and there are reports in the literature, many reports of using the IPT gene. I think it's very valuable, it's underutilized. And there are reports from the Voitis lab, for example, on using Wuschel and IPT to stimulate shoot formation. I think there's work that could be done there. And we have a new combination that we're working with that we're hoping to publish soon, that I think there isn't a magic bullet that's going to work in all crops. But we've got a new combination that I think is going to extend morphogenic genes into a number of new dicots.
Is a kind of cytokinin the signal, do you think?
Well, certainly, so for the use of IPT and Wuschel, you're looking for shoot proliferation. And so it is a cytokinin effect.
From the IPT gene.
From the
itself.
IPT gene, yes. And so that's one of the things that these morphogenic genes are doing is one of the first things that they allow us to do is to try to cut back on the amount of hormone that we have in the media. Because we've seen many times, we used to work with a gene called LEC2 LEC1 in maize, and it worked well for stimulating transformation in certain genotypes. We found that we could reduce the auxin tenfold. And this goes back to Greg's point. When we had the morphogenic genes and we had our high concentrations of hormones, it would give us aberrant morphologies. And so you really, you have to kind of rethink what you're doing when you're using these morphogenic genes. But the idea is to recapitulate normal development and to get them in and out of culture as quickly as possible.
Okay, so I think we're getting close to the end. I would like to thank again the panelists for this roundtable conversation. And of course, I would like also to thank you for joining us today. I know it has been a busy day starting from this morning. I just would like also to remind you that at 7 on the 2 room next door. We're going to have the welcome reception that is going on from 7 to 9. You're going to have the opportunity to see posters, the exhibitors, and then we're also going to start our silent auction. And since I see a slide that is coming up, we have an open survey that is open to both members and non-members. If we can go back to that slide. You have a QR code. Feel free to scan the QR code. It's a way for SIVB to really learn from you if what SIVB is offering right now is still relevant to you. And if it is not relevant, we really are asking you to help us to really envision tomorrow for the Society for In Vitro Biology. So scan the QR code, members or non-members, everybody can take the survey. It takes 5 minutes, 10 minutes the most. We really appreciate your feedback. You're going to see this slide coming through pretty much at the beginning of every session, but it is extremely important for us to be able to hear your voices so we can really build the next and future SIVB, okay? Thank you again, and I'll see you at the welcome reception tonight.